Now I'd like to introduce today's speaker, Dr. Taxiarchi. Dr. Taxiarchi is a postdoctoral research associate at Imperial College, London. She's a member of the research group of Dr. Andrea Crisanti, where a great deal of important research on gene-drive technologies and their potential application in the efforts to eliminate malaria has been conducted over the last decade. Crissa has worked on understanding mosquito gametogenesis and on the development of Cas enzyme-based genetic tools for population control. Her latest research publication was published in June and describes her use of protein inhibitors of Cas enzymes to create a gene drive brake system that blocks the spread of gene drives in mosquito populations. We're going to hear about some of that work today. So, Crissa, welcome, and thanks for joining us today. I'm going to give you the floor. Go ahead and share your presentation.
Dave already introduced. Today I will be talking about the ways we use to develop gene drives and a method to block them in Anopheles gambiae in our lab, in the Crisanti lab in London. To start with, why Anopheles gambiae? Because we're interested in eliminating malaria, and malaria is a disease that is still causing hundreds of millions of infections every year and hundreds of thousands of deaths every year, mainly affecting children under the age of 5. Now, the causative agent of malaria, which is the malaria parasite, is transmitted to human through the mosquito vector. So in order to remove the parasite from the equation, we can as well remove the mosquito vector from the equation of transmission. Now, this already has been done in the past very successfully in countries such as in the Mediterranean, where elimination of the mosquito populations through the use of insecticides led to a removal of malaria from those countries. And there are already we know 3,500 species of mosquitoes worldwide, and hundreds, more than 800 of those are in Africa, and only 3 species of them transmit almost 95% of malaria in Africa, and these belong to the Anopheles gambiae complex. So we are interested in those mosquitoes because we know that their ability to transmit malaria is genetically determined. So we can actually introduce a genetic modification that would potentially impair this ability to transmit, which is called vectorial capacity. This genetic modification could actually do that by either reducing fertility of the mosquitoes or increasing the insecticide susceptibility, introducing behavioral changes, or even introducing resistance to the parasite. Now, if a modification is introduced in a mosquito, how can that spread from few individuals into a larger population? Now, if we consider a modification in heterozygosis, normally, like all genetic traits, this modification will be transmitted to only half of the progeny of the modified mosquito, which is in heterozygosity, and then only in half of the progeny of that modified mosquito, and so on. And this way, it will not be able to increase in frequency above that release frequency, the initial one. So this way, just by introducing a genetic modification into a mosquito and releasing it into the wild, this will not allow for the modification to spread. But for example, how could we even spread a modification such as infertility? We can do that by the use of gene drives, and gene drives have this capacity to cheat Mendelian inheritance, and they can spread rapidly in a population. So a modified mosquito in heterozygosity will not only give the modification to half of its offspring, but actually to all of it. And this way, the modification can increase in frequency and spread in a bigger population. How does this molecularly happen? Naturally, gene drives, they have the ability to cut the DNA at the sequence, the DNA sequence where they're actually inserted in, and So yes, they have the ability to cut the DNA of the homologous chromosome of the sequence where they're inserted in, and they can actually this way allow themselves to copy across into the homologous chromosome, and this way they get duplicated in the genome and pass from heterozygosity to homozygosity. Now, any nuclease that is expressed specifically in the germ cells at the right time can actually act as a gene drive, and this can be anything from HEGs to CRISPR. So if we have a CRISPR, then we have Cas9 expressed from the gene drive cassette and a guide RNA that targets the actual insertion site on the homologous chromosome. They form a duplex that actually allows a cut in the homologous chromosome and then the copy of the gene drive. So there are two basic strategies that a gene drive can be used for, and this is population suppression or population replacement. This depends on the target of the gene drive and the cargo that it has. For example, the target could be a fertility gene, and the cargo could be resistant gene to the malaria parasite, which on one side will lead to population suppression or population replacement with, let's say, refractory to the parasite mosquitoes. So we focus on population suppression strategies, and for this reason, what we aim for in our lab is to actually generate transgenic mosquitoes, introduce a gene drive that in males, both in homozygosity and in heterozygosity, does not have any effect, any fitness effect. In females, in heterozygosity, has no fitness effect either, while in females in heterozygosity, so when the actual gene drive is present in two copies, then these females are not able to reproduce, and then they can lead to population crash. Of course, the aim is to also be able to transmit this modification, as we mentioned earlier, to a higher frequency than 50%, which is the Mendelian one. So targets that can actually create an effect like this, while males are unaffected and homozygous females are actually affected, of course, can be found in the sex determination pathway. So in insects, the sex determination follows a common theme. So we start with a chromosomal signal, which of course comes from the X, the presence of the Y, and then we have a master switch, a transformer complex, and at the bottom of this sex determination pathway lies the double sex gene, which will, in the end, determine whether a mosquito or another insect will be male or female. This is a transcription factor that has got two isoforms. So the double sex gene consists of 7 exons and 6 introns, and the two different isoforms, the male and the female isoform, differ only in the presence of the exon 5. So in males, the exon 5 is actually spliced out, while in females is present. So a very conserved sequence is the exon-intron boundary of this exon 5. And it is a very good target to for a gene drive, because a disruption of that would actually not allow the female isoform to be formed if the actual exon will not be included in the transcript. So seeing that this actual sequence is really highly conserved, not only among mosquitoes, but among different insect species, we went ahead and we thought we could actually target it and disrupt it, and see whether that will have the effect that I mentioned before, affect only the homozygous females. So we targeted this sequence, the splice acceptor sequence, using the CRISPR-Cas system. The guide RNA spanned the actual intron-exon boundary, and we introduced a GFP marker in the exon 5 sequence, which would allow us to visualize the knockout of that exon. And this way, the coding sequence of this exon is actually disrupted. And what we very interestingly and happily saw was that males, homozygous, XY individuals, homozygous for the disruption of exon 5, looked as normal females, as we expected. XX individuals that were heterozygous for the disruption looked like normal females. But individuals that genetically should be female because they have 2 copies of the X chromosome, but they actually have 2 copies of the gene drive, of the knockout cassette, do not look anything like females, but not even like males. They are they look like intersex. They do not bite, and they're sterile. So basically, we managed to obtain individuals that have that sort of phenotype that I mentioned earlier, since these individuals are completely sterile and also they do not bite, which is a pro because it's an extra advantage of having mosquitoes unable to bite and transmit the disease. So after creating that knockout, we went ahead and created a gene drive cassette to target this exon 5, which meant that we replaced the knockout construct with a cassette that carries the Cas9 gene under a germline promoter, which is in this case the ZPEG promoter, and a guide RNA targeting the sequence on the homologous chromosome under the U6 promoter, which is ubiquitously expressed. And this way, Cas9 is only expressed in the germline where we want the gene drive copying to happen, because in this way we can have heterozygous individuals in the soma, but only in the germline there will be homozygosity, which means that the homing will actually copy the gene drive cassette in the homologous chromosome, and all the gametes will be carrying the gene drive cassette, while the individual will still be heterozygous in the soma. So what we saw is that heterozygous individuals, both males and females, they are still fertile as we expected because their soma is unaffected by the disruption of the exon 5. But in the germline, homing does happen, and in this way, their transmission is up to 95 or even 99% in the offspring. So this was a great result, actually, which allowed us to then go and test whether this modification, having all these components that I mentioned before, affecting homozygous females, not affecting heterozygous females, being able to spread up to 99% in the offspring of the heterozygous individuals. We wanted to see whether we can actually have population suppression if we release these mosquitoes in a native population, in a wild-type population of mosquitoes. And what we did was a cage experiment, as we call it. So we introduced wild-type mosquitoes in a cage, and as well as gene drive males, heterozygous males, at 25% genotype frequency. And basically, by introducing those 600 mosquitoes in the cage, then we allowed them to, we blood fed them, allowed them to mate, blood fed them, and then retrieved their eggs, counted all of them, selected randomly 650 of those, screened the offspring, the larvae that hatched from those eggs, and counted them, and then introduced them again into the next cage. And this way, we have we have several generations that we follow up this population where we introduce these mosquitoes. And what we actually saw is that by targeting this intron-exon boundary in the double-sex gene can actually cause population crash, because what we saw is a rapid spread of the modification of the gene drive actually in the population. So from 25% of initial release frequency, we saw that the gene drive reached 100% frequency within 12 generations, and that actually agreed with the mathematical modeling that we performed before the actual release, before the cage experiment. So with blue and red, you can see a duplicate of this cage experiment showing how the gene drive modification spreads within the population through the generations. And in gray, you can actually see the model that agrees with what we saw experimentally. And of course, since the gene drive spread and it was introduced to 100% of the population, that led to a population suppression and crash within those 12 generations, since all the mosquitoes now are unable to mate and reproduce, since all the females are now intersex. Now, this shows that basically there's absolutely no resistance, at least within those generations and in this specific population in the cages, against the gene drive. And this means that basically we have a gene drive that not only can suppress a population quite rapidly, but also there's no resistance against it, while other gene drives, sometimes they reach a certain frequency, and then because of genetic resistance that arises against them, we see that the frequency of the gene drive then drops, and the gene drive sometimes gets completely removed from the population. So if we have a gene drive like this, how could we ever stop it in case we wanted to stop it? And in order to inhibit the spread of gene drives, we thought that potentially we can create a different kind of transgenic mosquito, a mosquito that we can call anti-drive mosquito, and we can introduce it into a population either against the population suppression or a population replacement. And what that would do would allow this anti-drive factor to spread in this population and remove the gene drive from it, allowing the wild type population to recover. So this was the aim. This is what we started thinking when we realized that we have a gene drive that is very very powerful. And we thought of using something that was naturally evolved to block Cas9, which we actually use in the gene drives we develop. So as CRISPR-Cas evolved in bacteria as immunity against the invading bacteriophages, bacteriophages also evolved their own immunity against the prokaryotic immunity, which are the AntiCRISPR proteins, the ACRs. And they're very small proteins that can inhibit Cas9 specifically. The ACR2A4 is one of them, as there are many different classes of anti-CRISPR proteins. But ACR2A4 has been shown previously to be a very strong inhibitor of SP-Cas9, which we use in our systems. It's been shown to bind with a very high affinity at the PAM recognition domain of the Cas9, and it binds to an assembled Cas9 guide RNA complex. And this way, basically, it mimics the DNA, and it doesn't allow the Cas9 to bind to the actual DNA target. So it impairs its own function, the function of the Cas9. So we thought, since we use CRISPR-Cas for our gene drives, if potentially we use genetically encoded ACR2A4 in the mosquito, and that is expressed in the same tissues as the Cas9, where the gene drive is, which means in the germline, then potentially we could block the binding of the Cas9 guide RNA complex because of the presence of the anti-CRISPR protein, and that wouldn't allow the Cas9 protein to cut the DNA and copy the gene drive across. And this would mean that there would be no homing of the gene drive. So theoretically, we started with this, and we thought in order to do that, we would need to genetically encode it into the mosquito. So we placed the ACR2A4 small protein, the gene of this protein, under the VASA promoter, which is another germline specific promoter that is active slightly earlier than the promoter that drives the Cas9 expression. And that is quite important because there have been some studies showing that it is important that there is some anti-CRISPR protein already present before the expression of the Cas9 protein, so that the inhibition is more efficient. And as well, we introduced an NLS signal to allow the export of the nuclear localization of the protein. And we introduced this cassette in a docking site in a mosquito, an autosomal docking site. So basically, by introducing that, we achieved expression of the ACR in the germline and only there. So we are not expecting this to be transmitted in a different way other than Mendelian, as this is just a normal modification that bears no gene drive and can only be transmitted at 50% of the offspring of a heterozygous mosquito. So moving ahead, we wanted to test whether basically the presence of this anti-drive, let's call it, can inhibit the progression of the gene drive and the homing activity. And what we did is, of course, we tested our gene drive for its super-Mendelian inheritance. We tested the anti-drive mosquitoes, but also we crossed gene drive with anti-drive mosquitoes so that these two modifications can be present in the same individuals. And what we would expect to see is homing inhibition by the small anti-CRISPR protein and normal Mendelian inheritance of both constructs. And when we actually performed this inheritance assay, we indeed confirmed that the double-sex modification is inhibited, it is transmitted with a very high rate. The anti-CRISPR modification is transmitted with Mendelian rules, while in the mosquitoes that carry both these constructs, we actually again see a normal Mendelian inheritance of the anti-CRISPR construct, but also normal Mendelian inheritance of the gene drive, the double-sex gene drive. And this actually shows 100% inhibition of gene drive homing, not just a slight inhibition. So what we actually understood from these assays, where we just allowed transgenic mosquitoes to mate with wild-type mosquitoes and see the inheritance of the constructs in their progeny, we saw that basically this anti-CRISPR protein is definitely expressed in the germline as we wanted it, and it acts. The activity of it is not inhibited by any factor in the mosquito. So these anti-CRISPR proteins can work very well within the germline of the mosquito, and they can block the Cas9 at 200%. So, of course, seeing this, we went ahead to test whether basically the introduction of anti-CRISPR mosquitoes into a population that is already invaded by gene-drive mosquitoes, whether that can actually stop the progression of the gene drive. So we went ahead to do cage experiment again, but this time we had a control cage, actually 2 control cages, as we performed a duplicate, where we introduced gene-drive mosquitoes, both males and females, to 50% of genotype frequency. And our experimental cages where there are also anti-drive mosquitoes present. So apart from 50% genotype frequency of gene-drive mosquitoes, we also introduced 20% anti-drive males in homozygosity. And again, we performed the same experimental method where we counted and screened the offspring and followed up this population of mosquitoes throughout several generations. And what we very interestingly saw, of course, the gene drive only cages followed the same pattern as before. So the modification spread rapidly in the population, and it reached 100% genotype frequency within 11 generations. Now, in the cages where we introduced the anti-drive mosquitoes, here you actually see the anti-drive genotype in green. Although the gene drive genotype managed to spread to quite a high frequency, it never reached 100%, and instead the AntiCRISPR genotype actually managed to spread as well to a degree that is above 50%, and we saw that the population never crashed. So definitely what we see from this experiment is that the anti-CRISPR, when the anti-CRISPR protein is expressed in the germline, it can actually not only inhibit the homing, but it will also prevent a gene drive-induced population crash in mosquitoes. And as you can see here from these graphs, where you can actually see the egg output, of course, in the gene drive only cages, we had a crash within these 12 generations. The egg output went down to 0, while in the experimental cages where the anti-drive was also present, the cage population was maintained at very low levels. So if we perform mathematical modeling to understand what it would look like after many generations of a similar release, we actually see that after 200 generations, the equilibrium between the drive and the anti-drive and the wild-type alleles remains stable. So there will be no removal of the gene drive, but no removal of the anti-drive either. So here, of course, there is a stochastic variation because this is stochastic modeling, but we actually see a very long-term equilibrium between these alleles. And the question was why? Why didn't the anti-CRISPR remove the gene drive from this population, since basically it can inhibit the Cas9 to 100%? We tested the fitness of the anti-drive individuals in heterozygosity and in homozygosity, and what we actually saw is that mosquitoes homozygous for the anti-gene drive insertion, and especially males, they had a higher fitness cost compared to wild type and compared to homozygous females, for example. And we saw that because of the larval output they produced, and because of their mating behavior, that they did not mate as well as wild type mosquitoes. So we wondered whether that was because of the presence of the anti-CRISPR protein or because of the the insertion, the genetic locus that we inserted, the AntiCRISPR cassette. And in order to understand that, we also tested mosquitoes that had a completely different cassette inserted in the same locus, and it was just a marker. And we saw that mosquitoes that carry this just simple marker in the same genetic locus also show some low fitness. So what we think after that is that basically is the fitness cost that we see affecting these anti-drive mosquitoes, and that we see also affecting the outcome of the inhibition of the gene drive, is mainly due to the fitness cost. And we modeled that, and you can see here a deterministic model of different fitness costs associated to the anti-gene drive construct. And the higher the fitness cost of the anti-drive constructs, of course, the lower the inhibition, and the harder it is to remove the gene drive from the population eventually. While if we increase the fitness of mosquitoes that carry the anti-gene drive constructs, then we see that the gene drive can actually be eventually removed from the population. So definitely the fitness cost of the anti-drive mosquitoes directly impacts the dynamics in the population. And if we manage to have a minimal fitness cost associated to it, we can increase the effectiveness of that. So we have already generated a new transgenic line that seems to be fitter than the one that we tested previously, and I showed you the results here. And we're already testing it in larger cages to understand the population dynamics in a more realistic, let's say, setting than the small cages we have in London. Yet what we can see through some different modeling is that even these mosquitoes that we generated and tested previously, and I showed you these results, although they do not remove the gene drive from the population, they could still stop it from crashing the population, even if the gene drive had no fitness cost. Basically, the fitness of the gene drive mosquitoes were equal to wild type, which is not the case actually. So of course the gene drive mosquitoes have some fitness cost, but even if that was equal to wild type, so there was no cost to that, our anti-CRISPR mosquitoes would be able to stop the gene drive from spreading and from crashing a population. And the good thing is that the gene drive does not even reach a very high allelic frequency, even if it's fully fit. So any stochastic variation potentially wouldn't allow it to crash the population. So this was all, and this is a work that has been done in the Crisanti lab, funded by the DARPA, and also partly by Bill and Melinda Gates Foundation. And I would like to thank the whole lab, the whole Crisanti lab, for all their work and help, and even fruitful discussions, and also our collaborators through the DARPA collaboration that we have with the Young Lab and the Beisel Lab and Polo GGB in Italy. Thank you very much.
Thanks a lot, Krista, for a really interesting and informative webinar. Really interesting stuff. And so I encourage our audience to submit their questions. We have some questions already. I'm going to go ahead and start with some of those. So one of them, one question had to do with the nature of the germline heterozygous alleles of the double-sex drive. What is the nature of the germline heterozygous allele of the double-sex drive? And this was from Pratima Trinuri down in Texas, I believe. But so was it drive NHEJ, or was it drive wild type? So she was interested in the heterozygotes.
Sorry. So the heterozygotes that we examined here, or sorry, the heterozygotes of the gene drive or the knockout?
Yeah, Pratima, maybe you can just chat in what you're in the chat box.
Maybe I can
We'll
probably
we'll pick it
see.
up when you do that. But in the meantime, let's go to another question. In the meantime, so Pratima, if you get a chance, go ahead and clarify. So Hector Kamada has a question. Do I understand correctly that if an anti-drive can eliminate a gene drive from a population, that the anti-drive, if it has a lower fitness compared to the wild type, will then itself disappear from the population?
So exactly, this would be the aim. So as I mentioned before, increasing the fitness of the anti-drive mosquitoes will allow them to compete with the gene-drive mosquitoes better. But the best thing to have would be sort of a fine tune between a high fitness and not so high fitness. So we don't want the anti-drive mosquitoes to be as fit as the wild type mosquitoes, because as Hector mentioned, we eventually want that modification to also be removed from the population. So balancing between a fitness higher than the gene drive, but also lower than the actual wild type would be the best to have for an anti-drive mosquito.
Yeah, and so just to follow up on that myself, so the relative fitness of the drive mosquitoes to wild type, of course, or at least the heterozygous drive is they're about equal. Is that right?
Um, Almost,
Almost.
not exactly equal, but almost.
Yeah.
Fitness is not high enough to sort of inhibit the spread of the gene drive, which is very powerful
Yep.
because of the high homing.
Yep. So you have to hit sort of a sweet spot in terms of the fitness levels of the
Exactly.
drive in order for it to be effective. So very interesting. Just going back to Pratima's question. So she was interested in the germline status of the heterozygotes.
Okay. Sorry, I didn't get that before. So basically, the heterozygous individuals that carry the gene drive. Let me go back to the slide. So it is interesting that I may find it while I'm speaking, but it is interesting that basically the double sex gene is required in the soma, but it is not required and not expressed in the germline of the mosquitoes. So while heterozygous mosquitoes in the soma will have no effect, because the disruption of only one copy will not cause any fitness effect. When those heterozygous individuals in the germline go through the homing reaction, they become homozygous, and they become homozygous, creating gametes that carry all the gene drive construct. Now that germline is still functioning absolutely fine, as you see from the fertility of those heterozygous mosquitoes carrying the gene drive. Probably you can't see it. I don't think I have a slide for that, but yes, they're fully fertile. If not fully fertile, almost fully fertile. And yeah, and they only produce gene drive gametes.
Yep. Okay. I think that got right to the heart of the question she was answering. So Summit Dole has a question. The ACR construct seems to increase when the gene drive is present at a high frequency in the population. Thinking of gene drive spreading in a spatial context, can the ACR construct stop the gene drive from spreading to new areas where the gene drive will be at low initial frequencies?
So that would require very complex mathematical modeling in order to answer the question. Potentially, what we could imagine is that antigen-derived mosquitoes could be used as sort of a barrier around a spot of release. Whether releasing antigen-derived mosquitoes in the same area and blocking the spread of gene-derived mosquitoes outside that area is something that we would be able to answer only after very, very complex mathematical modeling, which we don't have right now. But there is the idea behind potentially using the antigen-derived mosquitoes as a genetic barrier, let's say, for a more controlled release.
Mm-hmm. Yeah. So I guess the question is really getting at the idea of thresholds, and which was a question that I had as well in my mind, which was how many of these anti-derived mosquitoes would need to be released in a given population in order for you to have an effect. So I think with your modeling that you showed that if the fitness was good, then
Exactly.
you could start with I believe 25% is what your experimental population release was.
We started with 20% and
20%.
20, and with these, let's say, imperfect anti-derived mosquitoes, we managed to stop at least the population crash, not the spread. Yet, if the fitness becomes that the fine-tuned one that we mentioned earlier, potentially we could remove the gene drive by releasing the exact same frequency, 20%, whether 20% in a cage experiment or in a larger cage experiment. But whether that would be also feasible in nature is something that, again, would need to be modeled in a different way compared to what we have here.
Yep, great. Okay, let me go to another question from our audience. Let's see, Anna Romero Weaver has a question. I'm assuming that the wild type population of mosquitoes in a cage will all die at some point. So in your gene drive experiment, did you include a wild type control to prove that the crash in the population was indeed due to the gene drive and not to natural death of the mosquitoes? And if you include a control, do the gene drive populations crash before the wild type die? So this might have to, this might be maybe a clarification of the experimental design and how you progress from population to population.
Exactly. So it is not expected from a wild type cage to crash because of the way we perform this experiment. So we do not just release mosquitoes in one cage and then just wait for them to eventually die. But what we do is we take their offspring, all the eggs that they produce, and we select 650 eggs. Because in the beginning we introduced 600 mosquitoes, so we select 650 eggs, which in a wild type population should be fertile eggs, fertilized eggs that should have a very high I forget my words now, hatching rate, sorry, which means that at least more or less 600 larvae should hatch from those 650 eggs. And these larvae, once they grow, after they go through all the aquatic stages, are then reintroduced as adults in a new cage. And that cage, if we're talking about a wild type population, will have more or less 600 adults as well. And then from those 600 adults that we are allowed to mate, we blood feed them and get again all their eggs. And from all their eggs, we select 650 that should have a very high hatching rate. And from those 650 eggs, we take all the larvae that are produced and we go through cycles like this. So we don't just have one cage and we wait for it to die, but we create each generation is a different cage, basically. So in a normal wild type population, we are not expecting it to crash at any point.
Yusuk Lee has a comment. The population modeling is what I expected if anti-drive in Mendelian inheritance, but the actual cage experiment seems like anti-drive behaves more like a counter drive, increasing its frequency over time over the original drive.
Indeed, and that is not because of some drive mechanism that that the anti-drive has. That is solely because of the fitness. Let me find it. Because of the fitness advantage that it provides to the mosquito population. So this mosquito population is about to crash because of the gene drive, while when the anti-drive is present, it actually gives an evolutionary advantage to that population by inhibiting the spread of the gene drive. So this is why we see this increase in the frequency of the anti-drive, not because of any inherent driving mechanism within it.
Yep. Okay. I'm going to keep going here. Pratima has another follow-up here. So does the homing event produce no recordable functional or non-functional resistance alleles, especially functional resistance alleles that might evade the drive?
That's a very good question.
Yeah.
So I don't have this data here on this presentation, but of course we tested that both for the gene drive, but also the anti-drive. So both in the cages that of the original experiment, where we released the double-sex gene drive mosquitoes in the wild type population, but also in the cages where we released the anti-drive. And basically what we see is that there is some accumulation of mutations throughout these generations at the double-sex locus, but none of them is functional, and none of them produces an actual functional protein. So although there are some mutations there, none of them manages to exceed a threshold that would prevent the gene drive from spreading, and none of them actually produces an actual functional allele.
Okay. Yeah, good question. Okay, great. Here's a question from Jonathan Caliendo.
Sorry, before you go ahead, can I also add something? That as I mentioned before, we also tested for resistant alleles in the cages where we introduced the anti-gene drive mosquitoes, because obviously we wanted to rule out any ideas that could be, okay, what if suddenly in this cage there are resistant alleles, and what we see is not because of the anti-CRISPR mosquitoes, but because of some new kind of resistance arising. But what we saw is because the anti-CRISPR protein blocks Cas9 from cutting and eventually homing, we also have a reduction of those non-functional alleles that we see accumulating in our in cages without the anti-CRISPR mosquitoes.
Mm-hmm. Okay, good. Let me go on here. Yes, I was getting to Jonathan Calendo's question. Sure. The question is, so currently, if we were to deploy gene drive with no intention of crashing the wild type, the strategy would be to release it with anti-drive concurrently and hope they achieve some equilibrium to allow wild type to recover.
That's a very tricky question to answer, to be honest, because if we do not intend, I mean, generally the intention is to reduce the numbers of mosquitoes, to reduce the population size. And I cannot imagine eliminating, having an aim to eliminate the whole species. Yet there are gene drives built there that allow for the spread of the modification and reduction of the population, and then recovery of the population. And these are gene drives that that do accumulate resistant alleles within them. So if the intention of the release would be something, I don't know, a decision to be made by a different group of people. But I would imagine that the release of an anti-CRISPR mosquito could have that effect to keep the population in low levels and then potentially remove the gene drive and allow the population to recover, or release a gene drive that eventually will be wiped out because of resistance.
Okay, great. Let's see here. Do have some more questions, and we have time, so we're going to keep going, and encourage people to keep asking questions. We have till 12:30, and or if Krista drops over from answering them before then, we'll keep going. So here's a question from Qingjia Zhang. Sorry for my pronunciation, but since the release of the gene drive Mosquito could sometimes require repetitions over years, does this mean also a repetitive release of 20% anti-drive, or is it rather a one shot? Would the modeling result be the same or similar in the case of repetitive release? Okay.
So it depends. Okay, I guess to answer this question depends on what do we want to use the anti-gene drive for. Mm-hmm. So I guess if we're going for repetitive releases of gene drive, which again, releasing a gene drive aims for fewer releases compared to anything else, like a non-sustaining, let's say, strategy. So whether basically gene drive releases will be repeated has to do with the aim, the outcome that we want from the gene drive. So I guess if we need to do repetitive release of gene drive, is because we need more releases to reach a higher frequency of it. And in that case, I'm guessing we don't need to release anti-CRISPR mosquitoes at the same time, mm-hmm. because this way we're sort of like removing all the effort that we're doing through repetitive release of gene drives. I'm not sure I've understood this question clearly, but
mm-hmm. Yeah, it has to do, I think, more or less with the long-term. How this would play out over the long term, and I think you're probably not in a position really to answer it until some more modeling gets done.
Absolutely,
it seems like a lot of modeling is probably ongoing, and it would really inform a lot of this, I think. Mm-hmm. So yeah, okay. Coming down to our final questions and comments, but I'm going to go back to Yusuk Lee, who says, if anti-drive wants to achieve its goal, removing introduced gene drive, then do you need better fitness for the anti-drive alone, or are there other factors to be improved? So yeah, so good question about other things besides the fitness of the anti-drive mosquitoes that could affect that.
So this using these anti-CRISPR proteins can have a certain degree of inhibition, which means that since they block the Cas9 at the protein level, we're not talking about genetic removal of the Cas9 gene. We're talking about removal, let's say blocking of the protein. So in terms of this, what we're testing now is whether indeed only improvement of the fitness would allow the removal of the gene drive. This is something that we're currently testing. Yet, if we wanted to remove the gene drive in a different way, let's say through removal of the gene drive in the genetic form, then we're talking about completely different types of countermeasures, and that is something that I think will be discussed in the next webinar. This is potentially something that can be done by using nucleases, using Cas9 to remove the actual gene drive construct. That can have certain effects, that can have certain also risks, chromosomal rearrangements and all that. This is something that we're also testing in mosquitoes, but it's something like this has been also recently published in Drosophila and will be discussed in the next webinar. But while using the anti-CRISPR proteins and while blocking the Cas9 at the protein level, we hypothesized that increasing the fitness would be enough to remove the gene drive from the population, and we're currently testing it, but unfortunately we don't have that result yet, so we will figure it out.
Yeah, so are you thinking about the application of this kind of an approach? I mean, you mentioned it as a barrier, potentially as a barrier or as a containment measure for perhaps an outdoor field trial. Okay, is that how most of this is being envisioned, or do you see it as really being more of a countermeasure, really, as the last question was alluding to?
Surely it can be considered as a risk mitigation and management thing to use. Basically, it can be used to prevent any risks from, maybe, I don't know, I would say accidental releases or something like that, but also as potentially a countermeasure if needed in the future, which is something that I personally do not believe that would be needed in the case of mosquitoes and malaria control, but having a sort of a countermeasure and a way to stop a gene drive, which is quite powerful, is definitely a good risk mitigation and management thing to have.
Here's another question from Sumit Dole, who says, since the and it really has to do, I think, with more of the chemistry here of how these inhibitors work. Since the ACR blocks Cas9 produced from any location in the genome, would the same ACR construct work for blocking other gene drives that are not located at the double-sex locus? Or would you need a new ACR construct to be built for each drive?
That's a great question. And basically, this is another advantage, can be seen as an advantage or disadvantage of this method. Basically, any gene drive that uses Cas9, SP-Cas9, and doesn't have any specific mutations at the PAM recognition domain can be inhibited using these anti-CRISPR proteins. So there's no need for re-engineering of an anti-CRISPR construct in order to block a different gene drive if needed. It can be used against gene drives that target completely different locations in the genome and genes.
Would that be as long as they're using SPI-Cas9, or if they were using other Cas?
So every anti-CRISPR protein has a certain type of Cas that inhibits, and I'm not entirely sure if it's been shown that ACR2A4 can inhibit other types of Cas, but definitely the SP-Cas9 is one of them, and potentially it wouldn't be able to inhibit other types, other CRISPR types, but surely, or the unmodified, let's say, SP-Cas9 gene drives can be inhibited using this.
Yeah, I'll just give let folks know that are still here and listening that we're going to have a webinar from Amit Chowdhury from Harvard Medical School the week after next, the third in our series, where he does do a lot of work on these anti-CRISPR proteins, and this would be a great opportunity to come and hear more about, and he's interested in these not just from a gene drive perspective, but in other contexts as well. So if you're interested in hearing more about these anti-CRISPR proteins, there'll be more of it discussed at Amit's webinar. Recommend it. So I'm getting down to the end here. Jonathan Cando says, I guess the aim being suppression to be low enough numbers to break the malaria transmission, because elimination of transmission doesn't depend on eradication or crushing the population. Models tend to indicate that it can be achieved even with low numbers of vectors still present, which is a good point and one worth making. And Chrissy, you may want to just say something else about that.
Absolutely, no, absolutely. It is really important to point this out. It's really important to say that elimination of malaria doesn't mean that we have to completely wipe out mosquitoes. The transmission of malaria can be stopped with just low number of mosquitoes by treating people that carry the disease in the meantime. So basically, we're not solely expecting a gene drive to be let's say the one and only solution to the malaria problem. What we envision is a combination of different methods for malaria elimination, which includes drugs, vaccines, and this huge effort towards that. But definitely, if a gene drive is as powerful as the one that I showed you earlier, it can lead to a really good control of mosquito populations, hopefully, and in geographical areas that are not easy to reach, and potentially give vaccines and drugs and bed nets. So I think using the gene drive is giving this advantage of reaching those areas that potentially that are mainly affected by the disease, but really difficult to manage.